Hello everyone, so now we are going to look at the second types of mutation, which is chromosomal mutation. So in this lecture you will see the definition of chromosomal mutation, the two types of chromosomal mutation, we call it chromosomal aberration, and then chromosomal number mutation. We will also look at the types of chromosomal aberration, which there are 4 of them, and we will also have a look at the types of alteration of chromosomal number, which we have 2 types. Chromosomal mutation is defined as the mutation caused by alteration or changes of chromosomal structure, or in terms of the number. So under structure, which we sometimes also call aberration, we have 4 types. We have deletion, duplication, inversion, or translocation. So in terms of chromosomal number, we have 2 of them, which is aneuploidy and euploidy. Chromosomal aberration is defined as the rearrangement of a certain segment or parts of chromosome in one of these 4 ways. In deletion, 2 points on the chromosome will break off, causing the loss of a segment that contains 1 or more genes. So in this example, we have 9 genes. So in the point between C and D and the point between F and G will break off, and the DEF section will lose itself. So the point where the DEF was gone, which is the C, and the G will rejoin itself. So the product now is a much shorter chromosome containing only 6 genes, which are A, B, C, G, H, I. A popular example of this genetic disorder is what we call cri du chat. This is the French for cry like a cat. So this is caused by a loss of a small part of the short arm of chromosome 5. So here is the karyotype of a person with cri du chat. So as you can see, in chromosome number 5 over here, the small arms at the top here has lost a tiny bit of the structure. In duplication, it's very straightforward. This means that some regions of the chromosome has been copied or have been multiplied. So in this example, so again we have A, B, C, D, E, F, G. So in this case, the part where it contains gene F and G had been multiplied. So in the new chromosome, we have 2 copies of F as well as 2 copies of G. In chromosomal inversion, what happens is a segment of chromosome break off at 2 points, turn around 180 degrees within the chromosome itself, and then it will rejoin with the reverse orientation. So in our example here, it breaks off in between C and D as well as in between F and G. So the segment here that contains DEF will reverse in 180 degrees manner. So now we have a result of FED. So the segment that contains FED will rejoin into the chromosome in that particular order. So now we have ABC, FED, GHI. So the last one is called translocation. So translocation must involve the transfer of segments of chromosomes between non-homologous chromosomes. In a reciprocal translocation, so a segment of the blue chromosome is broken off and then transferred to the orange chromosome. So in exchange, the segment of orange chromosome is transferred to the blue chromosome. So there is no gain or loss genetic information, it's just transfer. In non-reciprocal, only one chromosome might gain or lose. So in this case, chromosome 4 has this segment broken off. So now the product is a much shorter chromosome 4. So the part that was broken off is transferred to chromosome 20. So now chromosome 20 has gained a segment of chromosome that came from chromosome 4. The second type of chromosomal mutation is in terms of the number. So the first type is what we call aneuploidy. So aneuploidy is a condition in which organisms possess an extra one chromosome or less one chromosome than the diploid number. So diploid is 2n. So 2n, so you can either gain 1 or you might have less 1. So the cause of aneuploidy is the famous non-disjunction that happens during meiosis. So it can happen in either sex chromosome or also in autosome. So this non-disjunction can produce gametes with abnormal chromosome. The normal gamete would have n. But obviously, if you gain one chromosome, you will write it as n plus 1. Or if you lose one chromosome, then you would write it as n minus 1. So in humans, for example, n is equal to 23. Therefore, if you have n plus 1, then you can just simply have 23 plus 1, which is 24. Or if you lose 1, then you have 23 minus 1, so that is 22. So this is a non-disjunction that might cause aneuploidy. So we have two scenarios here. The scenario number 1 is when non-disjunction happen in meiosis 1, and scenario 2 is non-disjunction that happens in meiosis 2. If non-disjunction happens in meiosis 1, then you will get something like this. Bivalent over here failed to separate like the bivalent at the top here. So the product, you will get two cells, two daughter cells that have 3 copies of chromosome over here on the left and 1 copy on the right over here. So during anaphase 2, when sister chromatids split, so now you will get 4 possible gametes. So half of the gametes will gain extra chromosome and the other half will lose 1 chromosome. And the one that is lost is obviously this one also. These are supposed to be there and this is supposed to be there. So this is the product of non-disjunction that happens in meiosis 1. If non-disjunction happens in meiosis 2, you will get slightly different result. So meiosis 1 was alright. So in meiosis 2, sister chromatids over here fail to split. So on this side, we get normal chromosome because the sister chromatids at the top here all successfully split. So we have 50% of normal chromosome. And then the other 50, we have 2 types. So either we have an extra chromosome over here, so we call it n plus 1, and the other one, sorry, that is 25%, and the other one is 25% also, loses 1 chromosome. Aneuploidy are very harmful to human. So we can look at the results in 2 different settings. So if aneuploidy happens in autosome or in sex chromosome. So in autosome, the most popular one is what we call monosomy 21. So monosomy 21 means we only have 1 chromosome in chromosome number 1. And the opposite to this is trisomy 21. The more popular name for this is Down syndrome. So this means we have 3 chromosomes at chromosome number 21. If aneuploidy happens in sex chromosome, we might have 5 different outcomes. So the first possible outcome is what we call the Klinefelter syndrome, where the genotype is XXY. These organisms are what we normally call female-like males. Second possibility is the supermale syndrome, which the genotype is XYY. We might also have superfemale, where the genotype is XXX, and we might have Turner syndrome, where the genotype is XO. So this is what we normally call male-like female. The final possibility is the opposite to Turner syndrome, which is if we have YO. However, a zygote or a fetus that has YO is normally never going to survive until adulthood, so it always results in the death of the fetus or miscarriage. So this is the karyotype of a person with monosomy 21. So as I said earlier, it means you have less one chromosome at chromosome 21. So here, this is the chromosome 21. So we're supposed to have another one over here, but it's missing. The common symptoms for individuals with monosomy 21 includes the short distance between eyes, over here, enlarged ears, and then contracted muscle. This is the karyotype of a person with trisomy 21, or the more popular name is Down syndrome. So if you look over here in chromosome 21, instead of only having 2, now we have an extra chromosome right over here. So the total chromosome for individual that has trisomy 21 is 47, and that is because 2n plus 1. So 2n in human equals to 46 plus 1, so we get 47. So some of the common symptoms include short stature, it means they are short in physical, flattened nose, protruding tongue, upward slanting eyes, short and broad hands, short fingers, and their growth, that includes their mental growth, are usually retarded. They are very prone to developing heart defects as well as gastrointestinal abnormalities, and then they normally have shorter lifespan. People with Down syndrome are normally sexually underdeveloped or sterile. The second type of chromosomal number alteration is what we call euploidy. So euploidy is involving the whole set of chromosomes. So just now we were looking at either +1 or -1. So in this case, whole set, which refers to n. So therefore, we can either gain n or we can lose n. So polyploidy refers to organism that possesses more than 2 complete chromosome sets. For example, we have 3n, so we call them triploid. So if we have 4n, we call them tetraploid. If we have 5n, we call them pentaploid. The cause of euploidy or polyploidy is self-fertilization between diploid gametes. So how did we get diploid gametes? Again, this is caused by non-disjunction. So as we can see here, one gamete that already had non-disjunction, and we get 2n, fused with another gamete that is also 2n, so the product is an embryo that is 4n, or we call it tetraploid. Another scenario might be a normal gamete that might fertilize with an abnormal gamete. So we have n, which is normal, it fused with a gamete that has 2n, so now we have a zygote that is triploid. The effects of euploidy can be devastating to humans. So it's very, very rare and basically impossible for us to find humans that have euploidy or polyploidy that survive until adulthood. However, in plants, they are actually beneficial. Euploidy in plants can be naturally occurring or purposely done by humans.